Good afternoon, and thank you for joining today's webinar on non-nutritive sweeteners hosted by the Food Forum. I'm Andy Zajac with FDA's Office of Food Additive Safety and a member of the webinar planning group, and I will be the moderator. For those that aren't familiar with the Food Forum was established in the early 1990s by the National Academies of Sciences, Engineering, and Medicine and the Food and Nutrition Board as a way of getting people from academia, industry, government, and other groups together to present information and perspectives on important food safety and health issues. The topic of a webinar on non-nutritive sweeteners was chosen by the Food Forum members. I've been told that at last count about 700 people registered for this webinar, so there seems to be plenty of interest in this topic. This webinar is scheduled for 1 hour and will focus specifically on the current and emerging science on potential health outcomes from non-nutritive sweetener consumption. Next slide, please. We are very pleased to have two excellent guest speakers today to present on this topic, Dr. Allison Sylvetsky of George Washington University and Dr. Frank Hu from Harvard's T.H. Chan School of Public Health. These speakers were invited by the Food Forum Webinar Planning Group and graciously accepted. After my introductory remarks, Dr. Sylvetsky will present a high-level overview of consumption trends of low-calorie sweeteners and current evidence from observational studies regarding health outcomes. Following her presentation, Dr. Hu will then discuss the state of science from meta-analysis and observational studies on consumption of low-calorie sweeteners and health effects, and touch upon future research needs and dietary recommendations. At the end of both presentations, there will be a few minutes to address questions that you have submitted. Just a reminder that your questions need to be submitted in writing through the WebEx. It would be best if participants submit their questions during the presentations as they occur to you rather than the end, so that we have time to organize the questions before the Q&A session. So let's get started. Next slide, please. Our first speaker is Dr. Allison Sylvetsky. Dr. Sylvetsky is an assistant professor in the Department of Exercise and Nutrition at George Washington University's Milken Institute School of Public Health. She also is director of the Bachelor of Science Nutrition Science degree program at GW. Her research interests include metabolic and health effects of low-calorie sweeteners and studying the role of dietary patterns in prevention and management of obesity and type 2 diabetes. Prior to joining the GW faculty in 2014, Dr. Sylvetsky did a postdoctoral fellowship in the Diabetes, Endocrinology, and Obesity branch at the National Institutes of Health, following completion of her doctorate in Nutrition and Health Science from Emory University. And with that, Dr. Sylvetsky, I turn it over to you.
Thank you for the introduction, Andy, and thank you all for tuning in to this webinar. I appreciate the opportunity to present. And I will specifically be discussing consumption trends and findings of mechanistic and observational studies. And so if it appears that I'm only presenting part of the story, that's intentional, because Dr. Frank Hu will be presenting on the randomized controlled trial results and future research needs. So low-calorie sweeteners are a topic of a lot of controversy, and they go by many names. So we'll be referring to them in this presentation as low-calorie sweeteners, but they're also known as artificial sweeteners, non-nutritive sweeteners, high-potency sweeteners, and sugar substitutes. And that's because these low-calorie sweeteners, which I'll give several examples of momentarily, are potently sweet and contain no or few calories. And in this presentation, we'll specifically be focusing on low-calorie sweeteners and will not be discussing sugar alcohols. Here are a few examples of common low-calorie sweeteners. Sucralose, commonly found as Splenda, saccharin, commonly found as Sweet and Low, riboside A, an extract of the stevioside plant, commonly found as Trivia, and then aspartame, commonly found as Equal. And you're likely familiar with these sweeteners, seeing them in the grocery store and on tabletops of restaurants. But what you might not realize is that while we call these low-calorie sweeteners, these represent a diverse class of compounds. And you can see there, looking at the structures, that these are a heterogeneous group of compounds that have very different chemical structures and different physical properties. And while many of the studies that we'll discuss evaluate these together, it's important to keep in mind that some of these effects may be sweetener specific, while others may be generalizable to the entire class of compounds if they're sweet taste receptor or sweet taste related. So there you can see sucralose, which is about 600 times more potently sweet than table sugar by weight. Saccharin to the right, 300 times more potently sweet than sucrose by weight. Rhodioxide A, an extract again of the stevia side plant, and then of the stevia plant, excuse me, and then aspartame, which is about 200 times sweeter than sucrose by weight. And what these have in common, of course, is that they're all potently sweet and are therefore used as replacements for added sugars in foods and beverages. It's also important to note that there have been numerous studies showing that these low-calorie sweeteners from a toxicological standpoint are safe and well-tolerated. And these studies have been conducted across species, across FDA-approved sweeteners, and at many dosages. So the focus of this presentation is not on safety, but rather on the effects of low-calorie sweeteners on weight and metabolic health. And part of the reason why this question of their effects on weight and metabolic health has become so important is because consumption of low-calorie sweeteners has been increasing in the United States. And this here is a figure from a recent paper that we published looking at trends in the consumption of low-calorie sweeteners using data from NHANES, the National Health and Nutrition Examination Survey. And what you can see here in blue are data in kids, in red are data in adults, and on the left-hand side of this figure shows trends in consumption of any food or beverage containing a low-calorie sweetener. So this is the percentage of people, the percentage of consumers who report consuming a food or beverage with a low-calorie sweetener on a given day. And if you look at the bars with the dashed lines, these were data that we reported in 1999-2000, or using data from NHANES in 1999-2000. And then in the solid bars are data from NHANES 2009 to 2012. And what you can see, particularly among children, is that the prevalence of consumption of low-calorie sweeteners has increased markedly over this time period. In 1999-2000, about 8% of children reported consuming a food or beverage containing a low-calorie sweetener, whereas in 2009 to 2012, this was over a quarter of children reporting a food or beverage containing low-calorie sweeteners. And we see similar trends in adults, though less dramatic. It's important to note that the NHANES methodology did change during this time period, going from a single 24-hour dietary recall to 2 dietary recalls, and therefore in the latter cycles, we were more likely to identify low-calorie sweetener consumers. However, this certainly doesn't explain the entirety of this increase. And if you look at the right-hand side of the figure, you can see that the majority of low-calorie sweetener consumption is from beverages, and the trends when we look at only prevalence of consumption of beverages containing low-calorie sweeteners, the trends are similar, again, more marked in children compared to adults. So beverages are the most commonly consumed source of low-calorie sweeteners, and the prevalence of low-calorie sweetener use tends to increase with age, family income, educational attainment, and is most common in individuals with obesity or who are overweight, and is also most common among females and those who identify as non-Hispanic white. But what's important is that the increases in low-calorie sweetener consumption are observed across all socio-demographic subgroups. So we see this in the general population as well as in different subgroups of the population. And these are some of the common sources of low-calorie sweeteners, the ones that most people think of: tabletop packets, diet beverages, and foods and beverages that are labeled as reduced sugar or sugar-free. However, more and more so, we see low-calorie sweeteners widespread in the food and beverage supply, oftentimes in products that people don't recognize as containing low-calorie sweetener. And these are just some examples of these products. They weren't selected for any other reason other than the fact that they contain a low-calorie sweetener. But the presence of low-calorie sweeteners isn't only limited to diet beverages. As you can see here, they're found in a range of different product categories. And given their widespread use and increasing consumption, there's been a lot of controversy over the extent to which these low-calorie sweeteners are helpful or harmful for weight management and the prevention of chronic disease. And this is a meta-analysis and systematic review that was published last year, and I'll show you a couple of the results of this review. So looking at the observational studies, and these are prospective cohort studies, the consumption of low-calorie sweeteners tends to be associated with more weight gain, and you can see that by looking at the estimates, which are all to the right of the center line. And this has also been shown in other meta-analyses This one assessing both body mass index and change in body weight in both kids and adults. And as you can see again, while the effect is modest, it does appear that consumption of low-calorie sweeteners is associated with higher body mass index, higher body weight. And in terms of other outcomes, similar findings have been reported in terms of diabetes risk, where the majority of prospective cohort studies show that consumption of low-calorie sweeteners is associated with higher onset of type 2 diabetes or higher risk of developing type 2 diabetes. And this has also been shown with other metabolic health outcomes, including cardiovascular outcomes, as well as fatty liver. And there's been an increasing interest in potential intergenerational effects. This was a study also published by Megan Azad's group, showing that mothers who reported consuming beverages containing low-calorie sweeteners, that their infants had a higher body mass index based on BMI Z-score and a higher risk of infant overweight at 1 year of age. And this was controlling for maternal total energy intake, diet quality, maternal pre-pregnancy BMI, and other relevant covariates. And therefore, there's been a question of could there be potential effects of early life exposure, whether that's in utero or during lactation. So how do we explain these epidemiologic findings? It could be that because these are observational studies, that what we're observing are effects of, residual confounding and reverse causality, inherent flaws of observational study designs. And it could be that we're seeing these because someone's already overweight, already struggling with their weight, or already following a poor diet, and therefore they're choosing to consume diet beverages as a way to stop a problem that's already, where the train's already left the station. But it's also likely that there could be various physiologic mechanisms that causally relate the intake of diet soda and low-calorie sweeteners to obesity, diabetes, cardiovascular disease, and related health outcomes. And what's most likely is that what we're seeing is actually a combination of both, that certainly reverse causality and residual confounding are at play, but there's also have been various physiologic mechanisms that have been proposed that may explain a causal link between low-calorie sweeteners and these adverse health outcomes. It's important to note, however, that these mechanisms have been predominantly shown in preclinical models, in vitro studies, and rodent studies, with very limited evidence in humans. And so some of these proposed mechanisms are shown here. I won't have time to go into great detail on any of them, but I will show you a few examples. One proposed mechanism is that sweet taste receptor mediated changes may be at play because these are all highly sweet compounds. Also, stimulation of adipogenesis leading to higher fat accumulation. Potential interactions of low-calorie sweeteners with the gut microbiota leading to alterations in nutrient absorption and leading to potential glucose intolerance. An impaired predictive relationship between sweet taste and calories because these compounds are sweet but contain no or few calories. And also a change in taste preferences and later dietary patterns that may result from exposure to these sweet compounds early in life. And again, most of these mechanisms have not been studied after chronic exposure in humans. So in terms of taste receptor signaling, the taste receptors, sweet taste receptors, are not only found in the oral cavity. We typically think of sweet taste as something that occurs in the mouth. And while it does, these taste receptors are also found in other parts of the body outside of the oral cavity. And there are sweet taste receptors in the pancreas, in the gut, and in the adipose tissue, as well as other parts of the body, although their functions are not well understood. And if you look at the right-hand side of this slide, you can see the signaling cascade for the heterodimeric G-coupled receptor, the sweet taste receptor, T1R2, T1R3. And when you have a sweet tasting compound, whether that's a caloric sugar like sucrose or a non-nutritive or low-calorie sweetener like sucralose, the sweet tastant binds to this receptor, starting a signaling cascade. And if this is in the mouth, a neurotransmitter is released that signals to the brain that we've had something sweet. And on the right-hand side of this diagram, there's an analogous cascade that occurs with sweet taste receptors in the gut, or for example, in the pancreas. Again, a sweet tastant, whether that's sucrose or sucralose, would activate this cascade. But in the gut, rather than this leading to a neurotransmitter telling the brain that we've had something sweet, this would instead result in the release of GLP-1 if this was an enteroendocrine L cell. Or if this is a pancreatic beta cell, this would result in the release of insulin. And this has been shown in vitro. And we've also done a little bit of work looking at this in humans. And while we can't say that this is a sweet taste receptor mediated effect, we and others have observed higher insulin levels following consumption of low-calorie sweeteners when they're given as a preload to an oral glucose tolerance test. So this is not low-calorie sweeteners in isolation. This is in combination or right before a glucose load. And again, this was acute exposure, and we can't say that this is sweet taste receptor mediated. But as Dr. Hu will be discussing, there have been several studies recently that have reported low-calorie sweeteners, specifically sucralose, after 4 weeks, leading to lower insulin sensitivity. Another mechanism is that these low-calorie sweeteners may stimulate adipogenesis. Again, these are effects that have been really only looked at in vitro, but the evidence from in vitro models show that when pre-adipocytes are incubated with low-calorie sweeteners, whether that's acesulfame potassium, saccharin, or sucralose, there's an increase in expression of adipogenic genes. So if this were to be the case in humans, this could promote fat storage and fat accumulation. Another mechanism that's been proposed is that there may be an impairment in the predictive relationship of sweetness and calories. And these studies have been largely published by the Sweeteners group out of Purdue, where rats are given unrestricted access to chow and then also yogurt. And on 3 days of the week, the yogurt is plain, unsweetened yogurt. And on the other 3 days of the week, the yogurt is sweetened with either glucose, a caloric sweetener, or saccharin, a low-calorie sweetener. So they get unsweetened yogurt 3 days of the week and sweetened yogurt 3 days of the week. And what they've shown repeatedly using different diets is that the rodents that are exposed to saccharin tend to eat more, gain more weight, and tend to develop relative glucose intolerance compared to the rodents that are given glucose. And so this is rodent data that is evidence for this potential impairment of the predictive relationship between sweetness and calories. However, it's important to note that other groups have not, there's another group that has not shown this using a similar design, and it's being challenged the extent to which this paradigm is applicable to humans. And it really hasn't been tested in humans, and this is something that needs to be done to see if we observe similar results in humans as have been shown in the rodents. Finally, another mechanism that has received quite a bit of attention as of late is that low-calorie sweeteners may affect the gut microbiome. And these data here are out of the Suez group from Israel, where they administered saccharin to 11 weeks, administered mice saccharin for 11 weeks, and then did oral glucose tolerance tests and showed that on the left-hand side of the figure, that the rodents who had received 11 weeks of saccharin exposure had a higher glycemic response during an oral glucose tolerance test compared to the rodents who had received water. And what was particularly interesting is then they did this again, but administered the water or saccharin with antibiotics, and the effect disappeared. And therefore, it could be that this is a gut microbiome mediated effect because the effect was no longer observed with the presence of antibiotics. And it's also important to note that these studies were also done with other low-calorie sweeteners, but the saccharin produced the most robust results, which is why these were the results that were ultimately reported. But this was shown with other low-calorie sweeteners as well. And to look further into the potential effects of low-calorie sweeteners on the gut microbiome, this group then did fecal transplant studies, where they took the microbiota from saccharin-exposed, glucose-exposed, or water-exposed mice and transplanted them into germ-free mice. And so the results that you see here are the glycemic responses of the recipients who were not directly exposed to low-calorie sweetener, glucose, or water. And so what you can see here in the blue is that the rodents who received microbiota from saccharin-exposed mice also had higher glycemic responses during an oral glucose tolerance test relative to the mice that had received microbiota from glucose-exposed mice. And so despite the fact that these mice did not directly consume the low-calorie sweetener or glucose, we're seeing similar effects, which suggest that the gut microbiota might be implicated in the impairments in glucose intolerance. But again, this has very limited evidence in humans. There have been a few studies that have assessed effects of low-calorie sweeteners on the human gut microbiome, and there are some in progress, but many have been cross-sectional or very small with significant limitations. And it's important to note on the right there that while I presented the results for saccharin, similar findings in terms of low-calorie sweeteners affecting the gut microbiota have also been shown with other low-calorie sweeteners. But again, these are all rodent data, and they need to be replicated in humans, and that's the big question. In humans, do low-calorie sweeteners affect the gut microbiota? And if so, what are the physiologic effects of this alteration? So to summarize, low-calorie sweetener consumption has increased over the past 2 decades, and foods and beverages containing low-calorie sweeteners are widespread in the food supply. And low-calorie sweeteners exert metabolic effects in vitro and in rodents, and acutely in humans. But the relevance to chronic exposure in humans is unclear, and I will now pass the mic to Dr. Hu will be talking more about the randomized control trials looking at chronic exposure in humans. So thank you, and I'll look forward to the question and answer session.
And let me introduce Dr. Hu. So we will move immediately on to his presentation. Dr. Hu wears quite a few hats. He is the chair of the Department of Nutrition and professor of nutrition and epidemiology at Harvard's T.H. Chan School of Public Health. He also is professor of medicine at Harvard Medical School and Brigham and Women's Hospital. Dr. Hu received his MD from Tongji Medical College in China and his PhD in epidemiology from the University of Illinois at Chicago. His research interests include nutritional and lifestyle epidemiology, obesity prevention, cardiometabolic diseases, and nutritional metabolomics. And with that, I turn it over to Dr. Hu.
Thank you, Andy, for your introduction. I want to thank Alison for her excellent presentation. My presentation is going to be focused on evidence from randomized controlled trials on the effects of low-calorie sweeteners, or LCS, on weight loss and metabolic outcomes. Okay, I'm going to I have some trouble forwarding my slides.
Frank, I can take over slides for you. Just tell me next slide.
Okay, yeah, next slide. I have no conflict of interest relevant to this presentation. Next slide. In the past several years, several meta-analyses have been conducted to summarize evidence from randomized clinical trials on the effects of LCS and body weight and body composition. Those meta-analyses have also summarized data from observational studies Alison presented. One of the most recent meta-analyses. It seems that all those meta-analyses have analyzed or summarized the same RCTs on low-calorie sweeteners body weight. The conclusions from those meta-analyses have not been entirely consistent. For example, this meta-analysis that was published in 2014 in the American Journal of Clinical Nutrition concluded that data from RCTs indicate that substituting LCS options for their regular calorie versions results in a modest weight loss and may be useful dietary tool to improve compliance with weight loss or weight maintenance plans. Next slide. In 2016, another meta-analysis that was published in International Journal of Obesity found that LCS do not increase energy intake or body weight, whether compared with caloric or non-caloric control conditions. Overall evidence indicates that the use of LCS in place of sugar in children and adults leads to reduced energy intake and body weight. Next slide. However, in the meta-analysis published in Canadian Medical Association Journal last year, it reached a very different conclusion. It states that evidence from RCTs does not clearly support the intended benefits of LCS for weight management. So the question is why different meta-analyses have reached a different conclusion, even though those meta-analyses are supposed to have summarized results from the same RCTs. Next slide. Let's look at those meta-analyses in a bit detail. This is the 2014 meta-analysis, which summarized results from 13 RCTs. The forest plot showed the point estimate for each of the trials, and the average effect size was quite modest. It's a less than 1 kilo weight loss a difference between the LCS group and the control group. This meta-analysis concluded that displacing sugar-sweetened beverages or SSBs or sugar with LCS options may result in modest weight loss. Next slide. This meta-analysis also summarized other health endpoints, including BMI, fat mass, and waist circumference. Those results are generally consistent with the results for weight loss. Next slide. In 2016, the International Journal of Obesity published a meta-analysis which summarized results from 9 longer-term intervention studies. And this meta-analysis also included a trial conducted among children. The effect size from this meta-analysis was somewhat larger. It's the weight, the difference in weight loss between the LCS group and the control group was 1.35 kilogram. This meta-analysis concluded that consumption of LCS instead of SSBs led to reduce the body weight. Next slide. And then the 2017 meta-analysis published in Canadian Medical Association Journal summarized results from 7 RCTs. This first plot showed RCTs with 5 RCTs with long caloric comparators, and the effect size was very small, is 0.17 kilogram, and was not statistically significant between the LCS group and the control group. This meta-analysis concluded that LCS was not associated with weight loss in the meta-analysis of 5 RCTs, except in long-term, 2 long-term trials with industry sponsorship. So it seems that the divergent conclusions from these 3 meta-analyses, to a large degree, are due to the inclusion and exclusion criteria used to select the studies to be included in the meta-analysis. And this meta-analysis analyzed on the subset of the RCTs that have been included in previous meta-analysis. In particular, this meta-analysis excluded 2 trials that were conducted in children. So this may explain some of the discrepancies in terms of the conclusions from those meta-analysis. Next slide. In the next few slides, I'm going to discuss some of the trials in more detail. Those trials are relatively large and also longer term. And this is a trial published in 1997 by George Blackburn. And this trial randomized 163 obese women who were enrolled in the weight loss program to 2 groups. One group consumed aspartame-containing products. The other group did not consume aspartame-containing products. In the initial weight loss phase, the 2 groups achieved the same amount of weight loss, about 10% weight loss from the initial body weight. And however, during the weight maintenance stage, the aspartame group had significantly less weight gain compared to the group who didn't consume aspartame-containing products. The authors concluded that this data suggests that participation in a new multidisciplinary weight control program that included aspartame may facilitate the long-term maintenance of reduced body weight. Next slide. Another longer-term trial was published in 2016 in obesity. This trial randomized 303 overweight and obese individuals who were regular LCS beverage consumers to two groups, water group and diet beverage group. And this study found that the individuals who were asked to consume LCS beverages, about 24 ounces per day, lost significantly more weight and maintained significant greater weight loss over 1 year than participants who were asked to stop LCS beverage consumption and consume water alone, and also about 24 ounces per day. The authors concluded that this result supports the use of LCS beverages to help with weight loss and maintenance. Next slide. In addition to those two longer-term trials, there were two trials that lasted for 6 months. And this trial was conducted in Denmark. It randomized 47 overweight participants to 4 groups, regular soda, milk, diet Cola, and water. This study found that daily SSB consumption, about 1 liter per day for 6 months, increased accumulation of liver fat, muscle fat, and also visceral fat compared with the groups who consume diet Cola, milk, or water. Next slide. In another 6 months trial, 318 adults who were regular SSB consumers were randomized to 3 groups: attention control, water intervention, and diet beverage intervention. After 6 months of intervention, the water and diet beverage groups significantly decreased energy intake from beverages. The diet beverage group had significantly lower amount, more weight loss, and water also better adherence. On the other hand, the water group had significant reduction in fasting glucose and improved hydration status compared with the control group. The authors concluded that replacing SSB with either diet beverages or water based on the consumer's preference and ability to adhere appears warranted at this stage of research. Next slide. In 2012, the New England Journal of Medicine published 2 trials conducted in children. In this trial, 641 normal weight Dutch children aged 4 to 12 years old were randomized to 2 groups. A group consumed SSB every day, and another group consumed diet beverage, which was sweetened by sucrose and ACE. After 1.5 year intervention, the diet beverage group had a significantly lower BMI Z-score, less body fat accumulation, and this study has several unique strengths. It is very large compared to other studies, and the duration of intervention is 1.5 years. And also, this is the only double-blinded intervention trial. The 2 groups, the soda cans look identical, so both of the children could not differentiate, could not tell whether they got the sugary beverage or diet beverage. Another advantage of this study is that urinary sucrose concentration was measured as a biomarker for adherence. Next slide. In another trial published in the same issue of New England Journal of Medicine, 224 overweight and obese adolescents who were regular SSB consumers were randomized to control group and intervention group. In the intervention group, the children received home delivery of bottled water and diet drinks as a replacement for SSBs. After 1 year of intervention, there is a significant lower BMI increase in the intervention group compared with the control group. The intervention discontinued after 1 year, and the children were followed for another year. At the end of the second year, the intervention effect disappeared. So this study suggests that home delivery of bottled water and diet drinks could be a useful way to improve home beverage environment and reduce consumption of SSBs. However, this study could not differentiate the effects of bottled water from diet drinks because they were delivered together to the homes of the participants. Next slide. A number of short-term studies have examined the effects of different types of LCS on glycemic control and insulin resistance. In this study, which was published recently in Journal of Nutrition, 100 healthy adults were randomized to daily beverage containing 0 milligrams, 350 milligram, and 1,050 milligram aspartame for 12 weeks. The authors did not find any significant effect of aspartame consumption on body weight, appetite, body composition, glucose, insulin, and the GI hormones. The authors concluded that findings do not indicate that using aspartame as a substitute for energy-yielding sweeteners would be problematic. Next slide. Also, in the recent published study in Journal of Nutrition, the authors found that daily consumption of 2 cans of beverage containing aspartame and SK over 12 weeks has no significant effect on insulin sensitivity and insulin secretion in non-diabetic adults. Next. On the other hand, a recent published study in American Journal of Clinical Nutrition found that sucrose consumption significantly decreases insulin sensitivity in healthy individuals. In particular, moderate consumption of sucrose over 14 days significantly decreased insulin sensitivity by about 18% versus only 3% reduction in the control group. So this study concluded that nutritional treatment of obesity and diabetes should focus on improving diet and increasing physical activity without promoting sweet-tasting foods, regardless of whether they contain caloric sweeteners or LCS. Next. Many studies have examined the effects of different types of LCS on postprandial glucose and insulin responses. In this study, preload of LCS, including stevia and aspartame, was used to replace sucrose, and it found that both stevia and aspartame significantly reduced postprandial glucose and insulin responses. And interestingly, the stevia group had the lowest postprandial glucose and insulin responses. This type of findings has not been widely replicated in other studies or other populations, and also it's difficult to generalize the acute effects of LCS on glucose and insulin responses to the health effects of long-term use. Next. As Alison discussed earlier, observational studies of LCS and body weight have many limitations. One of the most serious limitations is the reverse causation, because overweight and obese individuals tend to use LCS as a weight control strategy. One way to address the reverse causation in observational analysis is to look at changes in beverage consumption and the changes in body weight. Next. This is a study we published several years ago looking at changes in beverage intake and long-term weight gain in our 3 large cohort studies, the Nurses Health Study, Nurses Health Study 2, and the Health Professionals Follow-up Study. As expected, we found that increasing consumption of SSBs and fruit juices to a less extent was associated with increased weight gain. On the other hand, increasing consumption of plain water, coffee, to a less extent diet drinks was associated with lower amount of weight gain. So this result is more in line with the result from some of the RCTs that I presented earlier. Again, this type of analysis is very different from a typical observational analysis in which baseline consumption of diet beverages is associated with increased weight gain or a future risk of obesity. Here we're looking at changes in beverage consumption and changes in body weight, which to some degree mimic the design of a randomized intervention study. Next. In this analysis, we also estimated the effects of beverage substitution on long-term weight gain. We found that replacing SSBs or fruit juices with water, coffee, low-fat milk, tea, and also diet drinks was associated with significantly lower age-related weight gain. Next. Although RCTs typically provide stronger evidence than observational studies, there are many limitations of RCTs on LCS and weight control, including small sample size, short follow-up, decreased compliance, and increased dropout rates. And many of the intervention trials did not define interventions clearly. And also, there may be different effects for different types of LCS, and the effects may also be different between adults and children, and between healthy individuals and patients. Next. Based on current evidence, the 2015-2020 dietary guidelines recommend that replacing added sugars with LCS may reduce caloric intake and improve weight control in the short term, but little is known about the long-term effects. Next. In 2012, American Heart Association and the American Diabetes Association released a scientific statement on LCS. It states that there is some evidence that LCS could facilitate reductions in added sugar intake, decreasing total energy, and aiding with weight loss and control. However, potential benefits will not be fully realized if there is a compensatory increase in energy intake from other sources. Next. Recently, the American Heart Association released a scientific advisory on low LCS beverages and cardiometabolic health. The recommendations from the scientific advisory are very cautious, especially for children. It states that children should not drink LCS beverages long-term because the effects are unknown. For adults who are regular SSB consumers, LCS beverages may be a temporary strategy to reduce SSB consumption. Alternatives to LCS beverages and SSBs should be encouraged. Further research is needed on the effects of LCS beverages on weight control, cardiometabolic risk factors, and the risk of CVD and other chronic diseases. Next. So in summary, RCTs and observational studies of LCS and weight and metabolic outcomes have provided mixed and somewhat conflicting findings. There is some evidence from RCTs showing that replacing SSBs with LCS beverages could help in management of overweight and obesity, but longer-term and higher quality trials are needed. We also need more evidence on long-term effects of LCS consumption on cardiometabolic disease, cancer, mortality, and other health outcomes. And we also need to improve assessment of LCS intake by updating food bases to more accurately reflect LCS content in food and beverages and biomarkers to assess intake and compliance. Next. In terms of practical recommendations for adults, LCS can be consumed at a moderate level to replace SSBs in order to reduce energy intake and control weight. But water and other healthier alternatives should be recommended. For children, regular consumption of LCS is not recommended. We need longer-term RCTs to explore the effects of LCS by different types, by food, beverage sources, and also at different levels of intake and across different populations. Thank you very much for your attention.
Thank you, Dr. Hu, for your presentation. We will now move into the Q&A portion of this webinar, and for that, I turned over to Cypress Links of the National Academies.
Thank you, Andy. So for our first question, for both of our speakers, but we'll start with Dr. Silvetsky, what are the greatest limitations to assessing low-calorie sweetener consumption trends in the U.S. population today?
That's a great question, and there's a couple of major limitations. One is that in most of the food composition databases, for example, when we do our NHANES analyses, we use the FNDDS list of food codes, and they say, you know, a yogurt or a drink with low-calorie sweetener or a drink that's sugar-free, but it doesn't tell us what the sweetener is, and that's in part because the composition of foods and beverages is changing so rapidly. So we can't actually identify, using NHANES at least, and a lot of other studies as well, we can't identify the specific sweetener, just that something contains or does not contain a low-calorie sweetener. And in addition, a lot of the assessment of consumption relies on, obviously, self-report dietary assessment methods, and oftentimes people don't even know that the food that they're consuming contains a low-calorie sweetener. For example, if it's a high-fiber oatmeal that doesn't even say light or sugar-free, and only sometimes will the food composition databases actually pick up that it is something with a low-calorie sweetener. So issues with the specificity of sweeteners, issues with capturing a low-calorie sweetener is an issue with self-report for sure.
One thing I want to add is that in epidemiological studies, we need to explore the use of biomarkers as a more objective measurement of LCS consumption. Not all LCS have valid biomarkers at this point, but at least for sucrose, urinary concentration of sucrose has been used to assess intake and was adherence to interventions. So that's the area I think we need to invest more in terms of developing biomarkers for LCS consumption.
Great, thank you. One question we got in is specifically for Dr. Hu is, have you looked at individual types of low-calorie sweeteners in your cohorts for associations with weight?
Can you repeat the question?
Yeah. Have you looked at individual types of low-calorie sweeteners in your cohorts for associations with weight?
No, we didn't. We were not able to differentiate different type of LCS in our database because some of the sweeteners are more recent. Most of the diet drinks and also LCS in foods in the food supply is aspartame. So we assume that most of the LCS in our database is aspartame.
Great. Another question we received was, because there are sweet taste receptors in the gut and pancreas, among other places, is there investigation into low-calorie sweeteners as a risk factor for GI cancers as the prevalence is on the rise? For either speaker.
I can address that one. There are certainly observational studies, mostly cross-sectional, looking at cancer related outcomes, and there's been a couple, not with GI cancers in particular, but with lymphoma, leukemia, for example, showing increased risk with low-calorie sweetener consumption. But of course, reverse causality is a likely explanation there, as these are cross-sectional studies. But I'd like to point out again that these have been tested in terms of carcinogenicity and are believed to be safe at the dosages at which they're consumed. So there's not an immediate concern about cancer risk. However, because these may affect the gut microbiome, because these may, alter hormonal signaling in the gut, for example, certainly that's an area of interest, I would say, specifically with the gut microbiome potential, but I'm not aware of any studies specifically underway looking at consumption of low-calorie sweeteners in GI cancer.
Right. Yeah, I just want to add that in our cohorts, we look at the association between diet, beverages, and non-Hodgkin lymphoma and leukemia. The results are not very consistent. There is a positive association between diet, beverage consumption and increased risk of non-Hodgkin lymphoma in the health professional follow-up study, but not in the nurses' health study. Another very large cohort from NCI called AARP look at the association between diet, beverage consumption and the risk of those cancers, and they did not find association. So it's possible that the positive association observed in one of our cohorts that might be due to chance, but I think this is area we need much larger databases and large cohorts with longer duration follow-up so that we have sufficient power to analyze the relationship between diet, beverage consumption and a different type of sweeteners and risk of various cancers, especially some of the rare forms of cancers.
Great. Another question for Dr. Sylvetsky, and of course Dr. Hu if he wants to chime in. It was recently shown that low-calorie sweeteners are present in human breast milk. What are the potential implications of low-calorie sweetener exposure in infancy, and what work is being done to examine their potential effects during infancy and those critical developmental periods?
That's a great question, and that's an area that's of great interest to our group and others as well. And it is true that low-calorie sweeteners, we showed a couple of years ago, we published a paper showing the presence of low-calorie sweeteners in human breast milk, specifically saccharin, asulfame, potassium, and sucralose. And so that is an area of interest. I will say that the concentrations were very trace, so we don't know what the threshold of exposure would be to have effects. But of course, when someone's breastfeeding, they're getting potentially very low dose exposure, but repeatedly over days, weeks, months, et cetera. So really important area. And some of the potential implications that we're the most interested in are looking at effects on the infant gut microbiome, infant taste preferences, potentially affecting infant adipogenesis or infant, adiposity and weight trajectory, as well as their future taste preferences and dietary patterns. So I think a lot of the same mechanisms that I discussed that have been tested in rodent models would be specifically really important to test with early life exposure, given that infants are susceptible, the early life exposures we know are really important for future diet, future risk of chronic disease. So I think specifically gut microbiome, taste preferences, weight trajectory would be the areas that are most important to look into in terms of breast milk exposure.
Great. And I think we have time for one more question. And so if you could change one thing about current dietary advice around non-nutritive sweeteners, would you? And if so, what would it be? And we can start with Dr. Hu.
Well, the current recommendations on LCS are quite cautious. I think that's appropriate. The dietary guidelines and also the American Heart Association recommendations recommend a moderate consumption for adults who are regular SSB consumers. And for those adults, the preferred alternatives to SSBs should be water and other healthier options. But for people who want to have the sweet taste, and they can replace SSBs with LCS beverages, but at moderate level, and hopefully eventually the person can switch to water and other healthy alternatives. So to some degree, the diet beverages can be considered as a nicotine patch, which can help people transition from SSBs to a more healthy alternatives.
And I agree, and I would also add to that I think we need to have more emphasis on how low-calorie sweeteners are used, for what purpose, in what foods or beverages, because it's likely going to differ based on the context. And if people are just using low-calorie sweeteners in addition because, they have few or no calories, and therefore people think of them as, guilt-free, I can have 4 diet sodas and, it makes no difference. It's basically just, sweetened water is what a lot of people think. That is very different from someone who's very carefully replacing sugar-sweetened beverages with diet beverages. And so as a one-to-one replacement, replacing a sugar-sweetened beverage with a diet beverage, studies have shown that it could be at worst neutral, if not beneficial, for modest weight loss. But whether that's actually how they're used in the general population, given that we see low-calorie sweeteners in various food categories, oftentimes without knowing it, there needs to be more attention to where and how low-calorie sweeteners are being consumed, and then how they're being used and what the implications of those contextual factors might be. So I think it's very nuanced and not quite as simple as, are these helpful or not?
Fantastic. Thank you very much, and thank you to our audience for the questions. I'm going to turn it over to Andy to conclude our webinar.
All right, thank you, Cypress. So we're out of time, and this concludes today's webinar. And I would personally like to thank Dr. Sylvetsky and Dr. Hu for their excellent presentations and insights, and taking time from their busy schedules to participate in this webinar. I also thank those that submitted questions. When the webinar ends, you will be directed to a survey, and I would like to encourage all participants to fill it out so we can get your feedback about the webinar. And thanks again for joining us today.
Thank you.